Good afternoon. If I could ask everybody to take their seats, we're going to get started. And thank you so much for coming. This is an incredible turnout for mitochondria. Really a special welcome to all of our partners in research. I see some of you here, people who have included Salk in their estate plans. We are incredibly appreciative for you and all of your generous gifts. And for those of you who are just getting to know us, welcome, and we hope you enjoy learning about some of the incredible research that goes on in our labs here. I am Gerald Dean. I am Plan-Giving Counsel here at Salk, and I have had the privilege of working here for over 10 years and getting to know some of the fantastic science that goes on in our labs. It's incredible. I get to learn new stuff all the time because that's what they're doing. The discoveries that come out are truly remarkable, and as you'll see in just a few moments, really will be impacting all of our lives for the better. I'd simply like to remind you, while you are all here promptly, this is fantastic. We were oversubscribed today, and some people have been trying to RSVP as late as this afternoon. Please, in the future, so that we can best work our logistics, if you could RSVP by the date, the due date, if at all possible. We know things come up, but that really helps our events team out. So they asked me to remind you about that. Thank you. So before I introduce our scientist speaker today, I thought I'd show you a short video to just put this in a little bit of a broader context for some of the other research that we're doing in our optimal aging initiative. So I will have Kent show you our video.
One area that is emerging at the Salk Institute is our combined interest in aging and how we age as organisms. What's exciting about it for me is that we have between 10 and 15 existing faculty members whose programs are influenced or directed towards understanding how we age as individuals. Each one of these faculty members approaches the problem from a different perspective, and this allows us to work cooperatively together on the different systems that we are working on in such a way that we can unravel the small components and then put them back together to get a better understanding of how we age, why we age, and how this affects our cognitive functioning.
I'm excited as part of the initiative to work across disciplines with my colleagues in cancer biology or immunology to really bring them into neuroscience so we can make big discoveries.
Aging research at the Salk Institute has a long and successful tradition, and it started with establishment of the Glenn Center for Research on Aging more than a decade ago, and it has since continued through support by the Glenn Foundation and also through support by the American Heart Association and Allen Initiative that are both interested in furthering aging research at the Salk Institute.
Aging by far is the biggest risk factor for many common human diseases, heart disease, diabetes, obesity, cancer, Alzheimer's, and other neurodegenerative diseases. But what we don't understand is how aging is the biggest risk factor for each of those individual diseases. So I think a big push in aging research is to try to understand how different cells, tissues, and organs age in order to find the specific pathways in a person that has one or other of those particular diseases.
My lab is interested in understanding how individuals, their organs, and even individual cells within these organs age over the course of a lifetime. The chronological age, so the number that is on your birthday cake, is actually not very informative and doesn't tell us much about how you age. So there could be two 80-year-olds, one just qualifying for a Boston Marathon and the other one spending his or her days in assisted living facilities. And what we'd like to do is to be able to compare now individuals or organs and cells that either have a younger biological age than their chronological age or have an older biological age. And by comparing those two, we hope to identify underlying mechanisms that allow some people to maintain health throughout life, while others are not able to do that.
My lab works on telomeres and the DNA damage response. And we are optimistic that through our approach, by bringing different disciplines together and through the incorporation of computational approaches, that we will be in a unique situation to understand aging and to alleviate age-associated disease. Telomeres represent an intrinsic clock in our cells that only allow them to double a certain number of times, and thereby they prevent unlimited growth and immortality and, of course, cancer development.
My lab's particularly interested in the possibility that we'll be able to identify the factors that dictate how fast the ends of chromosomes erode, and potentially identify how efficiently those factors act in different individuals in a human population. And if so, the big pie in the sky goal is that maybe we could tweak the eroding process very early in life, where we could influence in the first decade of life, for example, how fast an individual's telomeres are eroding, so that late in their life they'll live well into their 90s without any of the pathophysiology of aging as the rest of us know it.
My research is actually directed at mitochondria, which are organelles inside most of your cells that actually convert the air that we breathe and the food that we eat into chemical energy called ATP. What my work has shown in that regard is that there's really quite a lot of data to support the idea that mitochondrial dysfunction is certainly involved in Alzheimer's disease, so we really need new directions and new research to figure out the underlying reasons for Alzheimer's disease, to find new therapeutic targets. By studying the basic biology of aging and understanding why aging is the biggest risk factor for Alzheimer's disease, such targets will be revealed.
So the work in my lab, we're trying to understand the brain and how it is that your brain changes with age, either in normal aging or in diseases like Alzheimer's. So you're probably familiar with cells in the brain called neurons, but what we work on are the other cells called glia that actually surround the neurons. And the reason we're excited about these cells is work we recently did showed that perhaps in normal aging and also neurodegeneration, these are the cells that change. And actually these changes in glial cells could be contributing to cognitive decline and the progression of all these different diseases. So as part of these bigger initiatives at Salk, what we're really excited to do is to work with the other groups and ask, well, can we target these glial cells or the changes that happen in aging and see if we can slow down the progression of cognitive decline or even prevent it from happening?
We're right on the cusp of solving some really fundamental problems that I think are going to change the course of how we age.
So, our optimal aging initiative, as named, is extremely ambitious, but we have incredible scientists here, so I have no doubt that you will be reading and hearing more about our really fantastic discoveries coming out of our laboratories within this initiative. And today, one of those researchers highlighted in our film, you're going to hear from directly. I am pleased to introduce Dr. Dr. Shadel. He's professor here at Salk and the Audrey Geisel Chair in Biomedical Science. He came to Salk via Yale School of Medicine, where he was a professor in the departments of pathology and genetics and was director of the Yale Center for Research on Aging. Dr. Shadel earned his BS a few years ago in chemistry from the University of Nevada, Las Vegas, received his PhD in biochemistry from Texas A&M, before being a fellow in the Department of Developmental Biology at Stanford. So his studies have taken him all over the United States. He's received many, many honors for his research, including the Amgen Outstanding Investigator Award, Breakthroughs in Gerontology Award, Glenn Award for research on biological mechanisms and aging. You don't want to hear me just keep on telling you about his awards. He's really smart. He's done lots of great stuff. And when he's not in the lab, he enjoys spending time with his wife, daughters, enjoys outdoors and music, and he's an all-around really nice guy. Help me welcome Dr. Gerald Shadel.
Well, thank you, Cheryl. That was a great introduction. I really appreciate it. And thank you all for coming. Wow, this is a packed house. I am really excited to talk in such an enthusiastic crowd. So first of all, thanks for all the support you're giving to the Salk and your interest in the science that's going on here. I came here 2 years ago, as Cheryl mentioned, from Yale. And one of the primary reasons I chose to move to Salk was I had been studying aging, but I really wanted to do more impactful aging research and really try to get into neurodegeneration research. And there's a strong history in neuroscience and neurodegeneration here at the Salk, and I thought this was my opportunity to marry those things together. And through this initiative, we're doing that. So almost as soon as I got here, this started developing. And let me turn my Wi-Fi off here. I'm going to get. And we got a huge grant that was talked about in that video, where multiple labs are together interacting to try to solve the role of aging in a major neurodegenerative disease, Alzheimer's disease. So I couldn't be more excited to have all of that just line up the way I planned it. That's like one of the few things that nothing ever goes as I plan it, so it actually surprised me. But really, it's an amazing place, and it's been the best move I've ever done is to be here. So today what I was going to do is talk to you first a little bit about this Salk Optimal Aging Initiative and unpack it a little bit from what you heard on the video. And then I'll tell you two short stories from my own laboratory about mitochondria and sort of a different way to think about how mitochondria might be involved in aging than sort of the traditional way. And so I hope that'll be interesting for you. So, I guess I need to start the show, huh? No. And we have great IT staff here, too. So as I already said on the video, age is in fact the biggest risk factor for most diseases, almost all diseases. And this graph really shows sort of the risk of death as a function of age of different diseases that you can get. And as you can see, some of them, like cancer and heart disease, sort of just steadily, the increased risk for that just steadily increases as you age. But others, like Alzheimer's disease and kidney disease, really take off after certain decades of life. So that just shows you that there's different aging trajectories for different types of pathology, and we really need to try to understand what it is about aging that is causing these types of increased risks at certain times in the lifespan of a person. And the promise of aging research really is, right now, the way medicine is practiced is it's all siloed. There's people who study, there's cardiologists, there's neurologists, et cetera, and each disease is treated individually, more or less. But if age is really the biggest risk factor for most diseases, if we could understand why and attack those pathways, we might be able to start preventing multiple diseases at once. And what that'll do will lead us to have live healthier longer, or increase what we call a health span. So the idea is not really to make everybody live to be 200 years old. It's to live healthier longer while we're alive. And the way we think we can do that is by understanding the pathways of aging and how those are intersecting with pathways of disease. So why is this such an important problem? Well, it's a huge problem in the United States and in the rest of the world. But in the United States, the baby boomers turned 65 in 2011. That's a lot of people. And so the number of people that are going to be 65 and older in 2050 is going to double, and the people that are going to be 85 and older is going to triple. So in other words, our demographic of our society is shifting towards a more aged population. And I just told you that aging is the biggest risk factor for those chronic diseases. That means chronic diseases are going to increase over the next 2 decades. And that's a big problem because it puts a huge burden, economical burden on the country. It causes soaring health costs. It puts burden on younger folks who have to take care of elderly folks. It limits the workforce availability. In other words, this is a huge socioeconomic problem. And something we need to really hit head on. And so that's the other reason I think this is critical right now to be studying aging research to try to combat this rise in chronic disease that's going to be coming up. So what about aging? So aging's complicated. That's one of the reasons we haven't solved it yet. And this is a really nice diagram showing that there are multiple different biological systems and mechanisms that we know are involved in aging, that go haywire with aging. And as was pointed out by Dr. Gage in the very first part of that video, many of us study these individual cogs in this wheel. But as this demonstrates, all of these are intersecting with each other, and one system can impact another. And so what we really need to do is sort of come out of our shells and start working with each other so that we can understand what the intersections are between these different aging pathways and how we can combat them. Furthermore, there's obviously environmental influences on aging as well: exercise, diet, toxins, pathogens, other fun stuff. So we need to understand all of these types of interactions if we're really going to try to solve this problem. And so just to point this out, and why Salk is a perfect place to do this, is if you look at the existing aging research at Salk, as was alluded to in that video, we have experts in most of those cogs in the wheel there. And so they're already here. And so what we need to do is work together to try to understand these intersections and to get a broader understanding of aging and how we might intervene. As Jan Karlsteder pointed out in the video, there is already a Glenn Center for the Biology of Aging funded by Paul Glenn Foundation. That's been here for 10 years or so. And as I already pointed out, there's a great strength in neuroscience, so I think there's a real opportunity here, and this is the thing we took advantage of first, is to really focus in on age-related neurodegeneration as our first target. And a year or so ago, we received a really nice grant from the Allen AHA Institute, which funded collaboration between 10 labs here at Salk. So you heard from 5 of them on the video. There's 5 more that were funded by this. And we're all working together to really try to attack the problem in the way I've already stated. That is, we want to look at the different hallmarks of aging, understand them in the context of an aging brain, and then understand how they're intersecting with each other in order to understand aging more comprehensively. And really, the breakthrough event that allowed us to really come together and culminate around this idea was from Dr. Gage's lab, where he was able to show that you can take skin cells from different aged people and directly convert them into different types of cells in the brain. For example, you can take fibroblasts and convert them directly to neurons, for example. And we've known how to do some of this stuff for a long time, actually, and the way we used to do it, you go through what's called an induced pluripotent stem cell stage. And when you do that, you sort of erase all the aging features of the cells. And so that's not good if you want to study aging, right? So what he was able to show that if you do this new direct conversion technique, you maintain a lot of the aging features of those cells, and now you can make an aged neuron and an aged astrocyte and an aged vasculature, et cetera. And if we can do that, and we're working on that, we can study the 5 systems in each individual cell type. We can mix those cell types together in what's called an organoid and ask how the different cell types interact with each other. And this generates a massive amount of data. And so we need computational methods and machine learning methods to try to understand the network of what's going on. And we have great biologists and computational biologists here that are going to tell us what it all means at some point. So that's sort of the broad goal. This is an 8-year grant. It's very ambitious. There's other aspects to it that I don't have time to get into. But this is really what we felt was a cornerstone or a nucleating event for a bigger initiative on aging, which we're calling the Optimal Aging Initiative. And so really what we want to do is expand this project, if that wasn't big enough, right? We're going to even expand it further. We want to understand the role of aging in other neurodegenerative diseases and other types of age-related brain pathology. So we want to make it bigger than it even is. We don't do anything small here at Salk. We just sort of go for it, right? And then the whole other aspect, which to be honest, we're still developing exactly how to go about this, but clearly it's fine to understand aging, but you really want to intervene and do something about it, as I stated, right? So we really want to have another arm of this initiative where we're trying to find interventions that can actually counteract some of these aging pathways and prevent disease pathology. So that's the goal, the bold idea. And in terms of those interventions, this is where we can sort of bring together many other people at the Institute who are working in other areas that are important for aging. For example, we have experts that are working on diet and circadian rhythm. I think Sachin Panda was the last speaker at this event, talking about fasting and how that can be beneficial if you do it right. We have people who are experts in metabolism and the microbiome, the bacteria that live inside your body and how that impacts your physiology. We have great immunologists who are working on inflammation and other immune-related factors. So we think these are points that we can intersect and intervene in aging, and we want to build out the initiative to target those pathways. And that takes advantage of existing centers here at Salk already. So we're looking, this is a broad initiative, and I think in the end, it could probably involve, I don't want to say all, but it could probably involve 75% of the investigators here at the Salk. All right, so then just to close this part out, and then get to mitochondria, because I know that's why you came, right? We sort of view it as a top-down approach, and that is, we're going to focus on the brain aging and the neurodegeneration, as I already stated first, and we think can be a template that's a huge problem. I mean, this is cognitive decline, it's neurodegeneration, this is a huge issue currently in the United States. But we want to use that information that we gain from that to ask how other parts of the body age as well. And I think we'll learn a lot from focusing on the brain first, but then I think it'll be applicable to other tissues and organs in the body. And then again, the idea is if we can understand that, we can stop fighting diseases one at a time and target these aging pathways as a way to more globally affect the body and have a huge benefit for people as we age, and so we can live healthier longer. So that's the goal. So we're passing the hat around now if you want to. No, I'm just kidding. Okay, so I'm going to switch gears now and talk a little science, if that's all right, and tell you about a couple of stories from my lab. And both of them have cute names, which I think will be entertaining for you. So, by the way, this was a good opportunity for me to show off the artwork of my daughter, my 13-year-old daughter, Jaydens, who made this. I actually asked her 2 days ago, I said, I'm commissioning you to make me a really cool piece of art that I can put in my talk. And she said, how much are you going to pay me? And I said, I'm going to feed you, so get to work. All right, so it goes back to this idea of these hallmarks of aging. And of course, one of them is what I work on, mitochondria. And you could argue it's the most important one. Rusty probably wouldn't agree with that. But he might, though, actually. He's coming around. He's actually kind of a mitochondrial guy now. So what are mitochondria? I'm sure most of you have at least heard of mitochondria. You might remember it from high school. You know what it does. What is it? What's this moniker? Well, that's a good one, but not a storehouse. That's like a bin or something. Right? The powerhouse of the cell, right? Come on, that's it. So what is it? So if you think about a cell's made up of many different functional units called organelles. And these are different membrane-bound or other large structures that are inside all of your cells that each have specific functions to help the cell work. In fact, you can think of it as it takes a village, right? So this, you can think of it as a village of organelles that are working together to make the cell work. So what do you need in a village? Well, first of all, you need manufacturing. You need to make stuff. So there are organelles that make proteins, there are organelles that make nucleic acids and things that you need for a cell to work. You need infrastructure. So there are railroad tracks and roads that run throughout the cell, that give the cell shape, that give it infrastructure, that give it, allow it, things to move around inside. You need distribution and global trade, so there are organelles that sort out components and move them to where they need to go, even send them outside. You have to have international trade too, so there's lots of things going on here. You need a trash can, so there are organelles that get rid of the trash. You need an IT unit, and so the nucleus, that's where the DNA is, right, where you get half from each of your parents, and that's sort of the blueprint for everything. But you got to keep the lights on, right? So you got to have something that's allowing the energy that you need to do all of this stuff, and that's what mitochondria are. And that's what we're going to talk about today. So what does it mean to be the powerhouse of the cell? What it actually means, mitochondria are, they have two membranes, and on this inner, really cool, folded up inner membrane here, there are large enzyme complexes in there. These are about 80 different protein subunits in these huge complexes. And what they do, what they do is when you're eating your food, that gets broken down into smaller constituents like amino acids, glucose, sugars, fatty acids, etc. Those get fed into the mitochondria, and the high energy electrons from that food get transduced down this system and extract the energy from those molecules. And what they do is they pump protons from one side of that membrane to the other. And protons are charged particles, they're positively charged. So if you separate charge across a membrane, you make a battery, right? And so you can utilize that battery, and we do. This enzyme here is called the ATP synthase. It zips those protons back through, extracts the energy from that, and makes this really high energy bond into what's called ATP, or adenosine triphosphate. ATP is the energy currency of your cell. It's used for everything you need to do, to think, to move your muscles, to fight infections, everything. And so you're eating and you're churning out ATP in order to do everything your cells need to do, your tissues need to do, and you need to do. And the key additional point is to oxidize your food, you need oxygen. And this is actually the reason you breathe. So this is the reason you eat and breathe, is you need the oxygen. That's the last place the electron goes in this chain. So you're breathing to fuel and fire up your mitochondria to oxidize your food to make energy, powerhouse of the cell. One issue with the oxygen dependency on this is the following. So normally what happens, it's a beautiful system. The electrons get put onto oxygen and they make water, no big deal, right? But unfortunately, sometimes the electrons hop off the bus a little bit early and get onto oxygen, and they can form a molecule called superoxide, and that can be converted to another molecule called hydrogen peroxide or hydroxyl radical. These are things that are called reactive oxygen species, or ROS. These are damaging molecules. They can damage your genetic information, they can damage your membranes, they can damage your protein, damage anything, essentially. And this leads to oxidative damage and oxidative stress, and this is a known way that when mitochondria start misbehaving, contribute to diseases and aging. So sort of a damage-mediated concept. And so, in fact, there is a long-held mitochondrial theory of aging. And the theory goes as follows. And so you have your normal mitochondrial metabolism that I just described to you, but you sometimes make ROS, and that ROS can cause damage. And if that gets out of control, you start to mess with the formation of those enzymes that are actually producing the ROS, and they make more ROS. All right, and if that keeps happening, now you got a cycle of that's going to lead to even more ROS, more damage, and you can imagine how this could fuel it forward and can be really a deleterious situation. And in fact, in the end, that can lead to an energy crisis in your cells, lots of damage, and even death of cells and tissues, and that's thought to be a driver of age-related pathology. You can also envision how environmental toxins, mitochondria are kind of a sponge for environmental toxins. Infections infect mitochondria in a different way. Inflammation, other disease states can actually impact anywhere along this chain here and exacerbate the situation. So that's sort of the standard mitochondrial free radical theory of aging. And of course, antioxidants are what you would do to get rid of this, because antioxidants get rid of those ROS and could be beneficial, right? They should be, right? Well, unfortunately, it's a little more complicated than that. And this is where my laboratory has contributed, that, life's never as simple as it seems at the very beginning. And there's actually a lot of controversy. There's a lot of evidence for this, and there's a lot of evidence that doesn't support this. And I'm going to show you something we discovered a few years ago that sort of contradicts this idea. And what we did is we were actually doing very fundamental research on yeast. Yeast is a single-cell eukaryotic organism. It has all those organelles I talked about, but you can actually use it not only to make bread and beer and champagne, etc., but to actually understand fundamental aspects of cellular aging. And what we actually showed was that if you give a yeast cell a little bit of mitochondrial stress and make a little bit of those reactive oxygen species, it actually extends their lifespan. Doesn't make it shorter. That's completely opposite of what you'd expect based on the mitochondrial theory of aging, right? So that's cool. What's going on? I'm going to talk to you about something called hormesis. So let's say if you are being exposed to any toxic substance, any poison, whatever, usually what happens is the more you take, the worse it is for you, right? There's a correlation between how much exposure you have and how much damage it's doing. But some compounds actually, at lower concentrations, have a beneficial effect before they have a toxic effect. This is called hormesis. And actually, Nietzsche actually knew about this. Because what doesn't kill you actually makes you stronger. But I don't read German, but I'm pretty sure there's a chapter in there of what is hormesis, mitohormesis. And so that discovery in yeast actually, and others had shown in other model organisms that similar things were happening. And so there's concept now of mitohormesis, that a little bit of mitochondrial stress can lead to adaptive cellular and systematic changes that are actually beneficial for you. And in yeast and other organisms can actually extend lifespan, i.e. are anti-aging, right? So we published a couple years ago that we wanted to know, so yeast is fine. I mean, nobody's going to write home tonight and say, wow, they made a yeast live longer. That's great. Maybe the wine industry or somebody would like that, but that's not what you're interested in. So we actually did an experiment in mice a few years ago to try to test out this theory. Is this happening in mammals? And if it's happening in lower mammals like mice, maybe it's happening in people, and this is something we can do. And so what we actually did is we spent like $40,000 and made a mouse that could actually allow you to do this. And what we did is we were able to give mitochondrial stress by knocking out an antioxidant enzyme inside mitochondria, inside a pregnant mouse. So we could cause a little bit of stress to the embryos inside the mom without her getting any stress. So we're only giving stress to the babies, the embryos. They weren't even babies then, they were just embryos. And some of them got stressed and some of them didn't. And then we let them be born, and they were no longer experiencing stress, and asked, were there any changes in their tissues, or was there any difference between these mice? And so the first thing we looked at was liver, and what we found was really super interesting, is that in the liver of the ones that saw the stress, they had more mitochondria. They had remodeled their mitochondria in a way that they're making fewer of those toxic reactive oxygen species, and they've upregulated cellular pathways that would normally get rid of reactive oxygen species. So that would be a mitohormetic or protective effect. And we took cells from those mice and actually showed that the ones that saw the stress actually were resistant to a second exposure to oxidative stress. So they were protected compared to ones that did not see the stress. What doesn't kill you makes you stronger. So we believe that these mitohormetic pathways are operating in mammals now. We want to understand the tissue specificity of it, and we also want to start aging these mice and see if they live healthier longer. And if they do, I think we'll try to figure out what the actual signal transduction pathways that are leading to that, and those are things we can target, hopefully, as a health span mediating therapeutic. All right, that's story number one.
The second one, I told you all have cute names. This one's mitoflammation now. Okay, so you're learning about mitohormesis, mitoflammation. You can see that I have a kind of a one-track mind when it comes to mitochondria. All right, so what do I mean by this? This is actually a really cool, equally cool story. And to start it off, I want to emphasize that there's something everyone knows, and that is with aging, there is low-grade chronic inflammation in your body. All right? And that inflammation is a contributor to many of the age-related diseases that we know are where age is the biggest risk factor. But nobody knows, actually, what the cause of this inflammation is. And so I'm going to give you one hypothesis. I have not proven this, but this is what we're working on. So I have to tell you another fun fact about mitochondria first, and that is mitochondria actually came from bacteria. So 2.5 billion years ago, which was right after I graduated from UNLV. People still argue about how this actually happened because we don't really know. But the idea was there was an ancient cell, and there's probably two different weird bacteria, in fact, that started commingling together and eventually decided that it was more beneficial to stick together than be apart, probably for metabolic reasons or because of the environment in the earth, and et cetera. But eventually, one of those bacteria became what we now have in us, which is mitochondria. So they actually came from bacteria. Well, bacteria are free-living single-cell organisms too. They don't have a nucleus, but they have DNA. And it turns out that your mitochondria have DNA too. So normally, when you think of DNA, I'm sure you think, oh, well, half came from mom, right? And half came from dad. But actually, a little bit more comes from mom. So your mitochondria actually have this circular mitochondrial genome that only comes from the mother. It's maternally inherited in mammals. And I encourage you to do this tonight. There is a really cool video called A Biologist's Mother's Day Song, where this guy does this beautiful song about all the things you get from your mom, and including mitochondria and other stuff from the egg, right? The egg's huge, the sperm's really small. Okay, so you get lots of stuff from your mom. But this DNA is the most important thing because it actually encodes genetic information and can cause problems. So what does that mitochondrial DNA do? Well, not surprisingly, that mitochondrial DNA has genes in it. It actually has 13 genes that are subunits of those complexes that make the ATP. All right, so a subset come from the mitochondrial DNA, see here. The rest come from the nucleus and get imported into the mitochondria to assemble with those. So the mitochondrial DNA is part of this energy transducing system. Well, DNA is DNA. I mean, if DNA gets mutated or that can be pathogenic, and mitochondrial DNA is no exception. In fact, there are now over 200 known pathogenic mutations in mitochondrial DNA that cause maternally inherited human diseases. All right, so this is a big area. I'm not going to go through, they're super complicated, but I don't need to go through that. But the other key point is that mitochondrial DNA damage and mutations also accumulate with age in you as you get older. So not only the mutations that if you inherited them, you're also gaining more mutations in the mitochondrial genome as you age. That's part and parcel of that mitochondrial theory of aging idea. And if you actually look at the pathology that's caused by those maternally inherited and other forms of mitochondrial diseases, if you look at the pathology they cause in aggregate, it actually overlaps quite remarkably with age-related pathology. So I think it fits this paradigm, or at least it's consistent with the idea that dysfunctional mitochondria could be involved in aging. But again, I'm going to tell you a completely different way I think mitochondrial DNA is maybe impacting aging, and that's as follows. We discovered a few years ago now that normally the mitochondrial DNA is all the way inside the mitochondria. Remember I told you there's two membranes, there's the purple, one outer membrane, and then there's this inner weirdo membrane. And the mitochondrial DNA is all the way inside. But we found a couple of circumstances where that DNA escapes. It gets outside of the mitochondria. And remember, this mitochondria came from bacteria, right? When that happens, the sensors that are in your cytoplasm that normally would detect a bacterial infection or a viral infection, which also have DNA, get activated and go, whoa, we're infected with something, right? But they're not. They're infected with mitochondrial DNA. All right, and what we showed is that when that happens, you activate these inflammatory genes in the nucleus. So there's a signal that goes from the mitochondria through the DNA all the way into the nucleus to activate inflammatory genes. And so now the organelles are talking to each other. We've got an interaction between the nucleus and mitochondria. That's a huge new area of biology of how all these organelles actually physically and functionally interact with each other. But the key point here for aging is you're causing inflammation from an endogenous source. And so that's what we think may be going on. I'll show you one movie of in red here is this is a cell with the mitochondria stained red, and green is the mitochondrial DNA stained. And if you look right here, you can see you'll see a DNA molecule escape the mitochondria. This one right here. It's almost out. So now it's out. It's just kind of like cruising around outside of mitochondria. And so that actually is what activates these inflammatory pathways in these cells. So the idea I want to leave you with with this last story is that, mitochondrial DNA we showed can activate these pro-inflammatory pathways. And so my add-on or alternative to the sort of classic mitochondrial theory of aging is that the mitochondrial DNA is not from damage and energetic deficits, although I'm sure that it's not mutually exclusive. But I think the mitochondrial DNA could be getting out and causing inflammation. And that could be part of the chronic low-grade inflammation that's happening with aging. If that's true, we need to figure out how to prevent that DNA from getting out or getting rid of it when it gets out or inhibiting the pathways that are being activated when it gets out. And those, again, I think could be potential targets for age-related pathology. And if you just sort of broaden this out, it's now been shown that the RNA from mitochondria gets out and activates different sensors. And again, they came from bacteria. There are other molecules in mitochondria that look like bacteria. And when your cell sees bacteria, it thinks, I'm infected, and does an inflammatory response. So I think there's a whole biology here that we're uncovering where mitochondria can contribute to inflammation in different ways. And of course, aging pathogens themselves affect mitochondria, and environmental stimuli, I think, is a way where these other factors could happen. So that's all I have to tell you today, other than now that we've uncovered this pathway, I have several really fruitful collaborations with other PIs here at Salk studying the role of this mitoflammation pathway in different contexts. So with my wife, Susan Keck, we're studying this in the context of melanoma. We're studying with Diana Hargraves. We have a new grant on colon cancer. I already told you about the Allen-HA initiative where we're looking at aging and Alzheimer's disease. And to really try to figure out how this DNA is getting out and what's going on, we're collaborating with a super talented investigator here, Uri Manor, who runs the biophotonics core here at Salk. So with that, I'll stop. I was told I had to stop at 15 after, so I got one more minute to tell another joke. But I want to end with the most important slide. None of the work any of us do could be accomplished without really dedicated students, postdocs, staff scientists, technicians, et cetera. And that's certainly true in my lab as well. And so I have an amazing group of people that I work with day to day who are super dedicated to science and super intelligent. And it's a pleasure to work with them. And then I've already talked about many of my collaborators, et cetera. So with that, I'd be happy to take any questions, and thank you for your attention.
Hi, I know that there's a lot of people who are interested in talking to Jerry Shadel. If you could wait until the mic comes to you so that everybody can hear, I'd appreciate it.
Do you see improvements coming from the development of pharmaceuticals, changes in the environment, or changes in behavior?
I think you hit all three of the most important nodes that we need to focus on. I think there are unique opportunities in all three of those spaces that could have huge effects on increasing health spans in humans. I think, one of the big, let's just talk about diet, which is a behavior, and an environmental influence on your genetics. It's been known for many, many years that caloric restriction extends lifespan in many, many different organisms that it's been tested in. So reducing the amount of food that you eat to a certain degree to where you're not starving, that will kill you, can actually lead to a but a hormetic type effect that can actually lead to beneficial adaptive changes that can be beneficial for health span. I think caloric restriction isn't that fun, and that's where Sachin's work here at the Salk is really interesting, because he's really trying to investigate intermittent fasting regimens where you eat only during certain times of the day, and that can really have an amazing impact on your metabolism, and he's shown that there's effects on mitochondria to this. So I think that's just one example. That's not pharmaceutical. That's changing your behavior, and when you eat can have a huge difference. Exercise. It's clear that there's a benefit to staying active and exercising, but I don't think we really fully understand what the pathways downstream of that are. And we have investigators at the Salk like Ron Evans, et cetera, who are really trying to figure out, what are the pathways that are activated by exercise and why are they beneficial? If you understand the pathways, then you have the chance for pharmaceutical intervention to try to tickle them, maybe without starving yourself, or maybe not running a marathon, but doing just a little bit of good exercise. So I think you're right on the money. I think all three. Environment, what would be a good environmental example? Don't smoke. Cleaner air, yeah. Any other questions?
2 questions, please. Going a few slides back, NADH, is that a nicotinic acid salt?
Which one?
It was about halfway through the presentation.
Oh, sorry. NADH?
Yes.
NADH is a nucleotide, a dinucleotide of adenine and nicotinamide.
Thank you.
Yeah.
And then your comments regarding alcohol in general, in terms of inflammation and stress, as it might be related to overall health, and the type of alcohol that you would prefer if you were a drinker.
Well, what answer do you want? I am a strong believer that alcohol is hormetic. So a little bit will eventually make you live longer, and I'm just joking. So interestingly, and this ties into NAD, and maybe this is the genesis of your question, but David Sinclair, many, many years ago, showed that there's an element in red wine called resveratrol, which actually activates an NAD-dependent enzyme and can have health span promoting effects. And so I wouldn't say go home and drink a ton of red wine, because there's so many other things that are deleterious about that, and the physiology on your liver, et cetera. But for sure, alcohol, chronic alcohol consumption is not good for you. It leads to liver fibrosis, it leads to fatty liver, it can even lead to liver cancer. So alcohol is not the route in which you want to try to extend your lifespan. But I think there are, the David Sinclair thing, I think, pointed to an important idea, and that is that you may be able to take certain metabolic activators that can have a very beneficial effect. But I don't think I'd do that through alcohol itself. And it does cause inflammation.
So recently I've seen the opening of clinics that are offering NAD therapy, and I'm curious what your opinion is of, and where the clinical research is in terms of NAD and its potential benefits.
Yeah, I mean, it's clear that NAD declines with age, and that in studies in mice, through the mechanism I just talked about, by activating these enzymes that are called sirtuins, actually has beneficial effects on age-related pathology, at least in model organisms. I think, there's a whole, I think it's Elysian Pharmaceuticals that's marketing NAD to take for this reason. I think the jury's still out on the beneficial effects in humans, and there hasn't been any really systematic clinical trial to assess the efficacy of NAD per se. But I think it's, it came out of basic biology of aging research. It has effects. I think it's still a viable area that we should be studying, and it's a potential way to have anti or health span promoting effects.
Are the clinical studies in humans going on right now?
I think so, yeah. I believe. Is it Rusty? Yeah. So the NAD, interesting in terms of what's going on in mitochondria, the NAD is actually made inside mitochondria when you turn NAD into NADH, and those are the high energy electrons that feed that chain to make ATP. So there are probably direct effects on mitochondria from NAD supplementation, but there are many other enzymes that require NAD. And the other important thing about NAD is it's a couple. It's a couple with another molecule called NADH, and the ratio of that is important too, because it's an indicator of the amount of basically oxidative stress or oxidant load that you have in your cells. And so it's tricky business to just add in one and hope the ratio stays the same. So I think there's still plenty of research to be done to figure out if that's going to work out or not. But it's on the table for sure.
I have a question about the mtDNA release.
Yeah, where are you?
Does that go across all ethnicities? So whether you're from Africa, Asia, India, Alaska, is it true for all humans?
I don't know the answer to that. In fact, what will speak to that, I think, is an understudied area with regard to the role of mitochondria in disease in general is that each of us have a specific type of mitochondrial genome that we inherit from our mom. We're not all identical, and different ethnic groups have different subgroups of what are called SNPs, different mutations in the mitochondrial genome that are slightly different in function. And it's really an area we don't understand well at all. It's like, what is the role of those in human disease, and I don't know about mtDNA release yet. This is brand new stuff. But this is left out. The scientific community tries to figure out what the genetic basis is for many different kinds of diseases. And they do what are called genome-wide association studies, where you look at different mutations and then put together. Guess what they throw away first? The mitochondrial genome. They don't even look at it and try to ask, are there associations between mutations or SNPs in the mitochondrial genome compared to the nuclear genome? It's completely ignored in the community, and I think that's a big problem, given the fact that mutations cause human disease, right? I don't know. In fact, there's a point mutation in mitochondria that causes diabetes, right? And so in a GWAS study for diabetes, I've never seen an association with mitochondrial genotype, ever. And I've asked people in front of them, why you don't, it's complicated, there's too many copies, I don't want to deal with it. But maybe I should do that.
Yeah, I have the mic here. I was wondering if there's any interaction between the mitochondrial system, the mitochondrial DNA, and the immune system. Can somehow the system detect mutations in mitochondrial DNA that have caused it to go bad and create antibodies to kill it off? And would the cells create new good mitochondria after the bad ones have been removed? And do you notice this in people who age well and people who age poorly? Is there a difference in their mitochondrial DNA?
No, that's a really terrific question. So what I showed you already was activation of the immune system by mitochondrial DNA. So if it gets out, it's binding to, that's an innate immune pathway that's being activated so that you know you're being infected by bacteria or viruses, et cetera. So we showed that there's activation of that immune pathway by mitochondrial DNA. But your question's a little bit different. You want to know if the mitochondrial DNA can produce something that's antigenic and can stimulate the adaptive immune system. And yes, the answer is yes, but there are pathways to prevent that from happening, or you'd be autoimmune all the time. But there have been a couple of studies by Heidi McBride in Canada who have shown that there is a mitochondrial antigen presentation system, and that this can contribute. But nobody's looked at it yet in terms of, does it contribute to aging or any disease state yet. But that's a really fantastic question, yeah. Got one behind you first. And
What
then
promotes mitophagy?
Promotes?
What promotes mitophagy?
Mitophagy. Well, so the standard answer to that question, you know, the main investigator who first identified mitophagy, the way they did it was to sort of poison the mitochondria to, you remember I told you it's like a battery, it develops that potential with the protons. If you poison that and collapse the battery, that's a signal for the mitochondria to go into the autophagy pathway, and there's thought to be specific receptors that recognize dysfunctional mitochondria, and that's the mitophagy pathway to get rid of defective mitochondria. Interestingly, two of the genes that get mutated in inherited Parkinson's disease are members of this mitophagy pathway. They're called Parkin and PINK1. And these are actually proteins that are on mitochondria that are part of that mitophagy pathway. And so there's data that, in fact, the mtDNA release that I showed you, that when you have defective mitophagy, you get more of that mtDNA release and activation of the inflammatory pathway. So it's a whole new way of thinking about maybe mitochondrial mediated inflammations involved in Parkinson's disease. But I think those are not the only signals for mitophagy. There's multiple different proteins that recognize different aspects of mitochondria that can drive it into the mitophagy pathway. But it's dysfunctional or damaged mitochondria are recognized to be gotten rid of so you can replace them with new ones, is the idea.
Have you studied whether or not anti-inflammatories medications have an effect on that breaking through of the DNA that triggers the inflammation?
We haven't yet. That's certainly something we want to do. We don't know if there's a feedback where inflammation causes more release and what's going on there. That's part of what we're trying to understand right now is how we can prevent it either after it's released or prevent the release altogether. And actually, we're still trying to figure out how it's getting out even. But yes, that's a viable avenue that we're looking into for sure.
Well, those were all really intriguing questions after a really fascinating talk. Dr. Shadel has agreed to stay with us during our reception because trying to keep everybody on schedule. I know that some of you don't like to drive during rush hour, so we respect that.
Who does?
But exactly. But you're all welcome to join us for a reception.